Now, hopefully, when I was talking about regulating transcription and controlling how often or how fast or in what tissues certain genes are activated, something in your head went, wait a minute, didn't he already teach us about that? There was something, those tissue-specific factors that bind to certain genes that allow them to be transcribed, and those are called transcription factors. And yeah, we did attend to that a little bit. So if you remember transcription factors, these are small proteins that float around inside the nucleus, and they only attach to the promoter region of genes that we actually want to transcribe. So this is a good way to make sure that only certain tissues are creating certain kinds of enzymes or certain kinds of proteins, because obviously you need a lot more contractile proteins in the muscle cells than you do in other types of cells, say skin cells. Now, the promoter region, of course, is just ahead of the gene, right? This is where RNA polymerase attaches. The transcription factors attach to the promoter region, but there's also this other part, and this is what I'm going to add in today. The enhancers. These are sections of DNA that are just a bit ahead of the promoter region, and it's the other thing that the transcription factors attach to. And these enhancers can either have a high affinity for these activator proteins and other transcription factors, or a low affinity. They can become active more often or active less often. And it is through a combination of these transcription factors, their attachment to the promoter, and their attachment to these enhancers that cells are able to take their DNA and regulate how fast or how often or even if certain proteins are being used. We talked about the operon in the last video. That's only for prokaryotes, but this occurs in eukaryotes as well. So in eukaryotes, activators seem to be more important than repressors. We can also have repressors, just like for the operon, on these enhancers, which shut them off entirely. But it seems like in eukaryotes, it's more important to have either the creation of these activator proteins, which turns on transcription for these types of genes, or not creating them, which would then prevent them from being transcribed. So you can see how it attaches here. The transcription factors attach to these enhancer regions, then they form this complex which then attaches to RNA polymerase. So when it begins transcribing, it's got the assistance of all of these proteins kind of moving along with it. All right, what else? RNA processing. Now, you may remember this slide from a previous video because we have discussed it before, but I'm going to add on a little bit to it. So, inside of your genes, inside of your DNA, you have your genes, and each gene is made up of coding regions of DNA, parts that will actually be coding and sending messages over to those ribosomes. Those are called exons, the active genes, the parts that are part of the message. And then they have parts that need to be cut out of the message called introns, right? So the introns get removed and left in the nucleus. The exons get kept in the RNA and exit the nucleus and then are expressed as proteins. So exons exit, exons are expressed, introns get left in the nucleus. And I create an analogy here to film editing, because when you shoot a movie, you have lots and lots and lots of film, right? And you got to cut out the deleted scenes. The introns are our deleted scenes here. So we already know that from earlier units. Exons, sections of RNA that code for proteins, introns, non-coding RNA. But in the process of splicing, and we get that completed RNA molecule, and obviously we add our cap and tail to it as well, we actually get some options about which exons we would like to include and which exons we do not want to include. We can customize our proteins by using this RNA processing step. So it might not be true that we get exon 1, 2, 3, and 4. We might get exon 1 and 2 and then exon 3, or exon 1, 2, and exon 4. We have alternative ways to splice it in order to create slightly different proteins with slightly different properties and affinities. So here is the machinery of the spliceosome. It's a series of RNA enzymes. So most enzymes are proteins, right? You didn't remember that wrong. But in this case, we actually do have some RNA molecules that are acting as enzymes. Those are called ribozymes. And a group of these ribozymes work together to form what we call the spliceosome inside the nucleus. The spliceosome will attach itself to an intron, the part we want to remove, and it will then bring together the two ends of the exons that we want to keep right next to each other, creating this hairpin loop right here inside of itself. That allows it to cut and splice in one move, removing the intron and keeping those two exons together. Right? That's how gene splicing works. So if we imagine a DNA molecule as a series of introns and exons, after transcription, alternative RNA splicing may generate two or more types of mRNA from the same transcript. In other words, we can create more than one kind of protein from the same gene. So you have 30,000 genes, but you actually have more options for making proteins than that because of this alternative RNA splicing. We can get different proteins from the same gene. In humans, more than 90% of protein coding genes appear to undergo alternative splicing. So this is not a niche example. This is how RNA splicing works in the vast majority of your genes, that it can make two or more different proteins by choosing which exons it's going to keep. So here's the example. So exons 1, 2, 3, 4, and 5 are all options for us to keep inside the final mRNA that we're going to make. And when we do the RNA splicing, we could have exons 1, 2, 3, and 5 together, or we could have 1, 2, 4, and 5 together, and these will make slightly different proteins. If you like, you can think of each exon as a functional part of the enzyme, and all it's doing is swapping out a few small parts of the enzyme which might have different kinds of functions. Maybe they attach to different sorts of molecules. And so they can keep most of the enzyme the same, but just modify it based on the cell's needs. Later stages of gene expression are also subject to regulation. The lifetime of an mRNA molecule helps determine how much of a protein is made, as do factors involved in translation. Okay, what does all that mean? Remember that the overall purpose of all of these mechanisms I'm talking about, alternative DNA splicing, chromatin packing, the euchromatin and heterochromatin, how tightly it's wound around the histone proteins, dark and light bands, the operons, the transcription factors with the enhancers, all of these things that we've been talking about are ways for the cell to determine how fast or how slow, or to turn on or to turn off transcription of certain genes, whether or not they're making certain cell products. So another way that they have, another tool in their toolkit for saying, hey, we don't need that much of this enzyme, we don't need that much of this product, or we need lots and lots of this product, is to change how long the mRNA molecule that's produced from a gene lasts. If it lasts a long time, mRNA can be used over and over and over again, so you'll get lots and lots of copies of the protein that it codes for. If it lasts a short time, then maybe you only get a couple of copies before it starts to break down. So by giving an mRNA molecule a short lifespan, you're going to get very few cellular products based on that, and maybe that's what you want. If you have a very long lifespan, you're going to get lots and lots of cellular products based on that RNA molecule, so lots of proteins of the same type, for example. A protein may need to be activated in some way, and eventually the cell will break it down. So when I talk about lifespans, the thing that determines whether it has a long lifespan or a short lifespan is going to be whether or not we're making a specific enzyme that targets this mRNA and breaks it down. I realize that this is starting to get very complex with terms that we learned in previous units, so if you need to go back and review that is fine. But one of the ways that proteins can target specific mRNA molecules, break them down so that we make less of that product, is by producing yet another kind of RNA, which is called miRNA, microRNA. And the microRNA is like a wanted poster for certain mRNA molecules. Let's say the cell wants to make an enzyme, but only a little bit of it, right? So the gene is transcribed, the mRNA is then sent to a ribosome to start being translated, but we only need a couple of copies of that enzyme. We don't want it to be copied over and over and over again. So what the cell will do is make a microRNA that is complementary with the mRNA that's been produced, and that microRNA will be attached to a protein which breaks down the target molecule. So basically, in order to shorten the lifespan of an mRNA molecule, the cell hires a hitman, and it gives them a little microRNA to find the target that it's looking for. The hitman is like an enzyme that breaks down RNA molecules. So here's what this would look like, all right? This pink RNA molecule right here, this is the one that's being used to make a certain enzyme. And we made it because we did need that enzyme, but we just don't want too much of it. So what the cell can do is make an miRNA, a microRNA, which attaches to part of that mRNA, and the protein it's attached to will then start chopping it up into little pieces, so it only has a very short lifespan. Another option that it has is to make an imperfect microRNA, so that it only partially binds to that mRNA. If it does this, it will not catalyze the process of breaking it down into little pieces. Instead, what will happen is translation will be blocked. So it basically slows down how much of this enzyme is being produced. It still has a relatively long lifespan because it's not being chopped up, but it slows down how fast translation can happen. So again, all of these mechanisms are either to modulate the rate of transcription and translation, or to modulate whether or not it's turned on at all, or to shorten the lifespan of how often you're creating these molecules. So to review all the different methods we have for control of transcription and translation, first, starting in the nucleus, chromatin, if you remember, is DNA and histone proteins. The DNA is wrapped around those histone proteins, and by wrapping it really, really tightly, creating heterochromatin, we can turn off any of the genes in that section of DNA. It just won't be transcribed. By lightening how much packing there is, by packing it looser, we are allowing those genes to be utilized by the cell. So that's the first kind of control that we have, is just how tightly it's wrapped around that histone, how tightly the DNA is packed. Once it's unpacked, we can actually use the DNA. We can transcribe it to make that mRNA molecule, and we get another couple of ways to control how often these genes are being used there. We can do it with promoters and enhancers and transcription factors that determine how fast and how often these genes are being transcribed. But once the gene is transcribed, we can also control what kinds of proteins are produced through alternative gene splicing. Once we have that mRNA molecule that we want and we send it out, we still have some options for controlling how fast or how often or how long that mRNA is being used and translated. By producing microRNAs, we can attach them to proteins, which will either stop translation, slow it down, or chop up that mRNA into little bits so it's only used for a short period of time and then it disappears from inside the cell. And even after we've made those polypeptides, we also have the ability to control how long they exist before we break those down as well. So the cell has lots of different methodologies for controlling how often or how fast or whether or not a gene is transcribed.